Hello, my name is Goreti Sales. I come from the University of Coimbra, formerly from the School of Engineering of the Polytechnic Institute of Porto. I am here to present you the work A Cellulose-based color test strip for equipment-free drug detection on site. Application to sulfadiazine in aquatic environment is the particular use that I bring you today. Antibiotics are very important to humans. In particular, they have been impacting humans' lives for the last 100 years, maybe. And from all the scientists that have been around this story, Fleming and Fourier are likely the most iconic ones. But many people have been striving still nowadays to engineer new antibiotics that can kill all bacteria around us that have a pathogenic effect in humans. Indeed, antibiotics have been the change of humans' lives throughout years, but we have been misusing them also. This includes overuse when we take them when we don't need, and this includes also using less than required regarding the infections that remain to be terminated because we didn't have the antibiotic until the end. And there is a third misuse of antibiotics that includes the overuse of these compounds in livestock and fish farming, which altogether are responsible for antibiotic resistance events that have been occurring worldwide. The use of antibiotics in livestock is incredibly great. It is difficult to imagine that 3/4 of antibiotics are being used in animals and not in humans. And this is a global effect. This is not a particular event of a specific country. So this means that antibiotics are being introduced in food and in the environment worldwide. Of course, this global introduction of antibiotics in the environment has led to antimicrobial resistance episodes. This, along with many other drivers, this is not a single one, but this is the one that I want to focus in this presentation. And I also want to take this opportunity to highlight that antimicrobial resistance is a global crisis, as mentioned by the United Nations, and this is time to action because we should do something about it. In this particular work, I want to bring you a new test strip that can be used to monitor antibiotics by farmers. And this would help farmers to use a reduced amount of antibiotic, and this, of course, with reduced costs. And this reduced amount should ensure that infection in the farmed animals are being cured. And also, if we use a reduced amount, the farmers will have lower cost and the environment will have less drugs being released into the environment. So this would be a win-win situation. The test strip that I bring you here is a very simple assembly that enables to monitor sulfadiazine on site. In terms of experimental activities, this is a cellulose-based test strip that has been oxidized by peridate in the first moment. Next, it is modified by an amine layer on top that is generated by the incubation of chitosan, and finally it is modified by copper because copper is able to form in the presence of sulfadiazine a pink color, and that distinguishes therefore the presence of this antibiotic. In terms of results and discussion, oxidation was the first stage of modification of the cellulose paper, and this was followed by FTIR studies. We followed in particular the carbonyl group being formed after the oxidation of cellulose, and we can see from this spectra, when compared to the blue one, which is cellulose with no oxidation, that any condition with peridate treatment was able to form a carbonyl group. However, this was more efficient when you use 60 degrees for 2 hours. The colors of the resulting papers are on the right-hand side, and we observed a little shrinking on the paper and a little yellowing, but this had no impact on the subsequent treatments. The next stage was the amide layer formation. We tried to improve this formation by mixing glutaraldehyde in different versions, but indeed ketosane yielded a stable layer of amine, and when sulfadiazine was added, we could see a pink color on top. On the contrary, when glutaraldehyde was present, some interference was observed with this reaction. So we ended up with a simple ketosane layer because it yielded a reproducible and stable color formation. The color being formed, indeed, is the color of a product of this reaction that is being leached from the paper strip into the solution. This product seems insoluble because it yields a turbid solution, and it also seems with a concentration dependency because the more concentrated solution of the three tubes is on the left side, and you can see that it has a more intense color. The selectivity study was done first with two other sulfonamide structures. This was sulfametazole and sulfamerazine, and one had a pink color and the other one had no color. Indeed, this could indicate that other sulfonamides may be tracked by the same test strip. Tetracycline yielded a yellow color and defloxacin had no color. But these two belong to other chemical groups, so eventually these groups do not interfere in the test strip results. Regarding the effect of sulfadiazine concentration, it was tested for a wide range of concentrations, and it was clear that the more concentrated sulfadiazine in the solution, the more color intensity was observed. The concentrations are increasing from the right to the left, and the test strip was dipped in the solution, and this was almost an immediate effect. And you can see the pink color being formed. Of course, this pink color is from an insoluble compound that is not homogeneously distributed, so the solutions may end up heterogeneous. As the calibration, we use the same concentration range and we try to add some linear correlation with the concentration. We searched for several possibilities, and the one that was most likely was to use the color from the picture that was generated from the liquid's air interface, and when we extract the red, green, blue coordinates of the RGB color systems of Windows, we can see there is a linear trend with the logarithm of sulfadiazine concentrations. When we use this principle to analyze spiked samples, it was very interesting to note that the experimental data generated from this analysis with a linear trend yielded errors that were more or less around 5%. So this confirmed the accuracy of the system. In conclusion, it is important to highlight that we understand that this is a suitable method for sulfadiazine monitoring in water. In particular, this is a very simple procedure to operate because you just need to dip the test strip inside a test tube of water. This is really inexpensive because the reagents are really inexpensive, and ketosane is also biocompatible and as well as cellulose. We have a very quick procedure because the reaction is immediate, and the results were confirmed accurate as well. There is also an environmental advantages for using this because this is a disposable test strip that has no or little significance in the environment. We'd like to acknowledge the funding institutions, which are FCT from Portugal, Compact 2020, and the European Regional Development Fund, and because they funded this particular project under the acronym of 3 Cs. I'd like also to acknowledge my team and to acknowledge you all that have been hearing this.